In this lesson, we are going to discuss the mechanisms of evolution. At the end of this video lesson, you should be able to discuss the different mechanisms of evolution and discuss how reproductive isolation leads to speciation. We may have first heard of evolution from Pokemon when we play this on our gadgets or trading cards, or when we watch this from our television screens. Evolution patterns of Pokemon became a common knowledge of those born from the late 90s to mid 2000s. For example, Bulbasaur has two evolutionary stages. It first evolves to Ivysaur. After reaching a specific level, it evolves to its final form, Venusaur. Pokemon gave us an idea that evolution is the change in the characteristics of a species over several generations. Evolution is defined as a pattern and process of changes found among species or organisms that are observed on several generations. For example, let us look at the evolution of vertebrates. A cladogram is a diagram used to represent a hypothetical relationship between groups of animals called a phylogeny. Organisms are said to be related to each other by having a common ancestor that had the same traits. For example, we can analyze the relationship of the following organisms using the following traits: vertebrae, bony skeleton, forelimbs, amniotic egg, hair, and eggs with shells. Organisms found before a trait or at the left of the trait in the cladogram do not have that trait. Organisms found after a trait or after the trait evolved from ancestors having the trait. For example, let us consider the bony skeleton. Sharks have vertebrae but do not have bony skeleton. Bony fishes, amphibians, primates, rodents and rabbits, reptiles and birds have bony skeleton. Evolution may not be easy to accept, especially considering the complexity and uniqueness of organisms. One may think that it is impossible for us to evolve from our said ancestors like the Australopithecus and the Homo erectus. But a misconception on evolution is when one thinks that evolution is a drastic change, like one species woke up the other day and that organism is a completely different organism. It is also a misconception that evolution works on the individual or species level. Evolution actually happens on the population level. Take note that genetic variations are the key to evolution, and these changes should be shared by a population and be passed on from one generation to another. Genetic variations would not lead to evolution if a single species or individual will not pass the changes to other species. Since evolution should happen on more individuals instead of only one individual, we should expect that evolution is a long process. In fact, we may not notice the changes that are already happening because these changes are happening in the genetic level. And these changes will only manifest on the next generation, which should also pass these changes to the succeeding generations. A population of species experiencing these genetic variations are said to be in the same gene pool. A gene pool is the collection of different genes within an interbreeding population. A gene pool is said to be evolving if its allele frequencies are changing through time, particularly from one generation to another. There are several mechanisms that lead to changes in the allele frequencies of the gene pool. These are mutation, natural selection, selective breeding, finite population, and gene flow. We're going to discuss each mechanism in this video lesson one by one. If all of these criteria are not met, the population is said to be in a Hardy-Weinberg equilibrium. This is a hypothetical population that is not evolving. Take note that this is only a hypothetical population since all populations are experiencing genetic variations. Let us look at how Hardy-Weinberg equilibrium works in populations. Suppose we have 10 individuals in this first generation of insects. Two phenotypes are existing, red and white. All red individuals are heterozygotes, and all white individuals have homozygous recessive alleles. To get the allele frequencies, we are going to count how many times the dominant and recessive alleles occurred in the entire population. The frequency of the dominant allele in the first generation is 7/20, and the frequency of the recessive allele is 13/20. Now, let's say after several times, the population already has its second generation of individuals. We may say that they increase in number individually, but evolution does not work that way. We should again look at the allele frequencies to look for genetic variations. For the second generation, the frequency of the dominant allele is still 7/20, while the frequency of the recessive allele is 13/20. This means that even if the population increased in number from one generation to another, it did not evolve as a species since their allele frequencies did not change. Hence, this population is in the Hardy-Weinberg equilibrium. And as mentioned, the allele frequencies of populations may be changed by any of the mechanisms of evolution. The first is mutation. Mutation is the random transformation of alleles or genes. The gene pool is modified if mutations alter alleles or if the entire genes are deleted or duplicated. Let us look at how mutation changes the allele frequencies of populations. This population of insect has a dominant allele frequency of 8/20 and a recessive allele frequency of 12/20. Looking closely at the effects of mutation on each level, the homozygous dominant allele mutated and became a heterozygote, although its phenotype did not change. This is an evidence that populations may be experiencing changes, but these changes are not felt or observed. Also, one heterozygote mutated to a homozygous recessive insect, changing the phenotype from red to white. These two mutations led to a loss of 2 dominant alleles and a gain of 2 recessive alleles. So how does mutation affect evolution? Mutation may give rise to harmful phenotypes which will affect the survival of the individuals in the population. A mutant allele may on rare occasions actually make its bearer better suited to the environment, enhancing reproductive success. A mutation is unpredictable. We will never accurately know which segments of the DNA will be altered or in what way. In multicellular organisms, only mutations in cell lines that produce gametes can be passed to the offspring. The next mechanism of evolution is natural selection. Natural selection is the differential success in the survival and reproduction of the species. Remember that the goal of all organisms is to survive and reproduce. However, the environment itself may be the threat to the survival of the species. Differences in the survival and reproductive success of individuals carrying different genotypes can alter allele frequencies. Suppose this population of insects are on a white ground and predator sees this population. The predator will more likely prey on the red individuals since it is easier to be seen due to its contrast from the ground. This makes the white phenotype suited for the environment. The death of 5 red individuals in this population made the dominant allele frequency to decrease and the recessive allele frequency to increase. Because of this, the white phenotype will be more likely to be more frequent on the next generations since it is helpful for the survival and the reproduction of the population. The third mechanism is selective mating or a non-random mating. If individuals mate preferentially with a subset of the population, such as their close relatives or what we call inbreeding, random mixing of the gametes does not occur, and genotype frequencies change. Sexual selection can result in sexual dimorphism, a difference between the two sexes in secondary sexual characteristics. These distinctions include differences in size, color, ornaments, and behavior. There are several ways on how organisms select mates. The first one is when individuals of one sex compete directly for mates of the opposite sex. This is called intersexual selection. In many species, intersexual selection occurs among males. For example, males fight for dominance in a group of females. This becomes beneficial to the next generation in such a way that the physical strength of the male will be passed on to that generation, allowing it to survive and reproduce. The other way of selective mating is through mate choice. In intersexual selection, individuals of one sex are highly selective in choosing mates. Most of the time, males display unique traits that attract female. This is where courtship rituals enter the scene. Males may exhibit unique appearances like when the peacock spreads his feather, and behaviors like when male's bird of paradise do a dancing ritual that should pass the standard of the female. This is important for some species because an attractive offspring may mean higher chances of reproduction, thus, higher chances of survival. The attraction formed between the organisms may be classified as assortative or disassortative. For assortative selection, An individual prefers an individual with the same phenotype. For example, a red-footed female bird would want a red-footed female bird. On the other hand, for this assortative selection, an individual prefers an individual with a different phenotype. For example, some humans like that their partners have the opposite height because it complements their height. Now, let us discuss the fourth mechanism, which is a finite population. A non-infinite population will lead to higher fluctuations of allele frequencies. This means that genetic variations are more evident if there is a limited number of individuals in the population. We can use the analogy of flipping a coin. The smaller the number of coin flips, the more likely it is that chance alone will cause a deviation from the predicted result. These limited populations would also lead to genetic drift or chance events that fluctuate allele frequencies unpredictably. This means that individuals may expect any changes in the environment, but they are not really in control of their own survival and reproduction. This simply goes that genetic drift is like a lottery of survival and reproduction. We have two types of genetic drift. Suppose we have a population of 10 hibiscus. There are 5 red hibiscus, 4 pink hibiscus, and 1 white hibiscus. However, 4 red and 1 pink hibiscus plants were stepped on by children. This makes the reproducing population reduce to 3 pink, 1 red, and 1 white hibiscus plants. Since there are more pink reproducing flowers, we can now expect that there would be more pink flowers in the next generation. However, in the second generation, only 2 red flowers were allowed to reproduce. This leads to a conclusion that on the third generation, all plants will be red pure breeds. This is what we call the founder effect. This happens when there is an isolation from a larger population that forms a new population whose gene pool differs from the original. In this example, an allele is lost from the gene pool, but it is a matter of chance that the allele for white is lost and not the allele for red. The other type of genetic drift is what we call the bottleneck effect. A sudden change in the environment such as fire or flood may drastically reduce the size of a population. A severe drop in population size can cause the bottleneck effect. In this example, let's say that there are 3 variations, red, green, and blue. These individuals are in a bottle. The shaking of the bottle is the bottleneck event that would unpredictably dictate which individuals will survive. At the end, only 4 red, 1 green, and 1 blue comprise the surviving population. In bottleneck effect, certain alleles may be overrepresented among the survivors, others may be underrepresented, and some may be absent altogether. In summary, genetic drift is significant in smaller populations. It can cause allele frequencies to change at random, leading to a loss of genetic variation within populations. However, genetic drift can cause harmful alleles to become fixed. The last mechanism of evolution is gene flow. Gene flow is the movement of genes into or out of the population due to the movement of either the individuals or gametes. Suppose we have 2 separated populations of insects. One is homozygous dominant for red, the other is homozygous recessive for white. If barriers between the 2 populations are not present, the individuals may interact and reproduce, causing a gene flow. Because alleles are exchanged between populations, gene flow tends to reduce the genetic differences between populations. In fact, if it is extensive enough, gene flow can result in 2 populations combining into a single population with a common gene pool. However, the lack of gene flow leads to speciation. This means that since 2 populations cannot allow an exchange of genes, the 2 populations are said to be of different species. Speciation is the process by which one species splits into 2 or more species. This is a speciation event in which there is a divergence between 2 distinct populations. This means that they are highly related, but they are not entirely the same. For gene flow to occur, 2 related species should mate. Mating would then lead to fertilization. Fertilization has a goal to give rise to viable and fertile offspring. However, there are some barriers in which these 3 important events are not met. Differences in habitat, mating time, and behaviors like courtship rituals may hinder mating. Differences in genitals and gametes may hinder fertilization. If fertilization is not met due to the barriers are said to be prezygotic. This means that the reproductive barriers occurred before a successful fertilization. However, there are cases wherein fertilization is successful, but the offspring are at risk. The offspring may either have reduced viability, reduced fertility, or hybrid breakdown. These are what we call post-zygotic barriers. Let us look at an example for each of the isolation barriers. For geographic isolation, two separate species within the same region, even though they are not separated by obvious physical barriers like mountain ranges, rarely come across each other at all. Two species of garter snakes in the genus Thamnopus occur in the same geographic areas, but one lives mainly in water while the other is primarily terrestrial. For temporal isolation, species that breed at various periods of day, seasons, or years may not be able to combine their gametes. In North America, the geographic ranges of the western spotted skunk and the eastern spotted skunk overlap, but the western spotted skunk mates in late summer and eastern spotted skunk mates in late winter. Also within closely related species, courtship practices that attract mates and other activities specific to a species are efficient reproductive barriers. Such behavioral rituals, a way to recognize possible mates of the same species, allow mate identification. Blue-footed boobies in Galapagos mate only after a courtship display unique to their species. Part of the script calls for the male to high step, a behavior that calls the female's attention to his bright blue feet. In mechanical isolation, mating is attempted, but morphological differences prevent its successful completion. For example, the shells of two species of snails in the genus Bradybaena spiral in different directions. Moving inward to the center, one spirals in a clockwise direction and the other in a counterclockwise direction. As a result, the snail's genital openings indicated by the arrows are not aligned, and mating cannot be completed. The eggs of another species may not be able to fertilize the sperm of one species. For example, sperm may not be able to live in the females of the other species' reproductive tract, or biochemical processes may inhibit sperm from entering the membrane covering of the eggs of the other species. For example, sea urchins release their sperm and eggs in the surrounding water, where they attach and become zygotes. Gametes of various animals, such as the red and purple sea urchins seen here, find it impossible to unite because proteins attach very poorly to each other on the surfaces of the eggs and sperm. Now, let us look at the post-zygotic barriers. For reduced viability, genes of different parent species can interfere in ways that hinder the production or survival of the hybrid in its environment. Some salamander subspecies of the genus Ensatina live in the same regions and habitats, where they occasionally hybridize. But most of the hybrids do not complete development, and those that do are frayed. If the hybrids are vigorous, they may be sterile. If the chromosomes of the two parent species vary in number or structure, the hybrid meiosis can fail to produce normal gametes. Since infertile hybrids cannot produce offspring when they mate with any parent species, genes cannot freely flow between the species. And lastly, some first-generation hybrids are viable and fertile, but when they pair with one another or with any parent species, the offspring of the next generation are frail or sterile. Strains of cultivated rice, in the process of their evolution from the common ancestor, acquire distinct mutant recessive alleles at two loci. Hybrids between them are vigorous and fertile, but the next generation plants that bear so many of these recessive alleles are weak and sterile. Although these rice strains are not yet known to be distinct species, they have started to be distinguished by post-zygotic barriers. To conclude this lesson, let us review the following key points. Evolution is the passing down of heritable traits from a generation of a population to another generation. Mechanisms of evolution include mutation, sexual selection, natural selection, non-infinite population, and gene flow. And lastly, the absence of gene flow leads to speciation. Speciation may be due to prezygotic or postzygotic reproductive barriers. And that ends our discussion on the mechanisms of evolution.